give you a little overview of how enzyme catalysis is used for carbon capture here. Just to scope my talk a little bit, I will start by saying that I'm going to talk about what we call free enzymes. So free enzymes means that that the enzymes are manufactured in a big fermenter and separated from the cells that have produced them, and then used in a cell-free setup. So that's different from, for instance, the so-called wholesale catalysis, where the microorganism is actually a part of the process. But the application of free enzymes is pretty substantial, as you see here. Let's see. I was hoping I was getting a marker. Sorry about this. Well, yeah, here. So these are some of the applications that runs this industry. It's about 4 or 6 times smaller than the production of chemical catalysts, but it's still an industry. And perhaps more interesting, we can talk about what's on the horizon for this type of application. And of course, today I'm going to talk about carbon capture and how enzymes can facilitate that process. But the other upcoming areas are, as you mentioned, Jiwang, are biomass and plastic waste, which can be depolymerized by enzymes. And these industries are more or less on the verge of starting. There's a handful of biomass plants that does enzyme catalysis for the breakdown of biomass, and I think the first plastic bioprocessing full-size plant will start in a year and a half from now. So all these are quite big industries over here, and carbon capture alone, at least that's what the company says, has a transformative capacity for the industry. So the scale of carbon capture can be so big that it will massively change that industry if they can develop a competitive technology. So obviously, there's a very big interest in that. Now, the reason that we are interested in enzymes, of course, is that they're exceptional with respect to both specificity and catalytic efficacy. And that comes from the fact that they have a 3-dimensional active site, like the example over here, which can make a network of interactions with both the substrate and the transition state that lowers the energy of those and hence provides both specificity and catalytic turnover at very high rates. But there are a large number of limitations, and a lot of the research in this field is about limiting those problems. And the biggest problem is that enzymes are typically physically unstable, so it is often hard to use them in an industrial process. But if we zoom in on carbon capture, there are different types of enzymes that are used. By far, the biggest area here is enzymes that accelerate the absorption. And the most prominently, that's an enzyme called carbonic anhydrase, and that's what I'm going to talk mostly about today. But before I return to that, let me just mention that there are also other free enzymes that are used in carbon capture. And for example, oxidoreductases can be used to reduce CO2. So instead of just capturing it, you can also actually convert it. And the most well-investigated enzyme in this area is format dehydrogenase, which reduces CO2 to formic acid, and that requires obviously electrons. So therefore, this is a much more complicated process. You need either electrons from an electrode or from a mediator so to run the process. But it's something that's coming of age also. Finally, I have one example down here, which I believe is very important, but it's almost unexplored, which is enzyme catalyzed desorption. So there are enzymes like carbamases and esterases and others that can break down the complexes, the carbamates that Jeff was also talking about, and hence they can accelerate the stripping during scrubbing processes. And then actually very often these processes are limited by the desorption rate. So it would be very interesting to see if we could improve that through enzyme catalysis, but it hasn't been done yet. Right, so let's return to the acceleration of adsorption, and to point out the basics of that. I made this sketch here, which has 2 major steps. So if you want to capture carbon dioxide in an aqueous solvent, you have one equilibrium here between the gaseous compound in the mixture and the dissolved carbon dioxide in the linear form, and then that is converted to the bicarbonate ion, and the enzyme that we're working with, the carbonic anhydrase, or CA for short, is catalyzing that reaction. It's important to notice that both of these reactions are equilibria. So carbonic anhydrase, like any enzyme, catalyzes the reaction in both directions. In many cases, we neglect that, not for this reaction, but for other enzyme reactions, because the energy landscape is such that the backwards reaction is very slow. But for this reaction, both the reactions are very important. So it's important to keep in mind that Ca is not producing bicarbonate quickly, but it's equilibrating this acid-base reaction quickly. Talking about quickly, the kinetics is such that the uncatalyzed reaction proceeds about with this rate constant. So aqueous CO2 in water has a half-life of about 20 seconds or something, and then it's converted to bicarbonate. Now the enzyme increases that to a much higher rate. As a matter of fact, the rate is about 7 orders of magnitude higher. At least under optimal conditions, the enzyme can increase the rate by that big a factor. So that's it's very fast turnover with sometimes up to 1 million turnovers per second. So it's very effective. I shouldn't mention though that this is not a very high acceleration. Actually, it's in the low end of what we see for enzymes. Typically 10 to 15 orders of magnitude is what enzymes can do. So this is not a particularly proficient enzyme or anything. Now, to understand why the equilibration of this process here helps the capture, of course, the first and most straightforward thing is that it prevents a buildup of aqueous carbon dioxide. So when the carbon dioxide comes from the flue gas or other sources, the concentration here increases, and the enzyme makes sure that it's quickly converted to the soluble ion, and hence it prevents that the CO2 steams back into the air phase. But that's only part of the effect of the Ca. To realize the effect more, we have to consider the interaction between the CO2 and the sorbents. Of course, we don't absorb CO2 in pure water, but in a solution. And the mechanism of Ca depends very much on which sorbent we're using. If, for instance, we're using a primary amine, as is often done, like here, then we've formed the type of carbamate ion that Jeff was talking about. But it's interesting to notice that the substrate for the reactant for forming that is actually CO2 and not bicarbonate. So it's not an advantage if the enzyme removes the CO2 and converts it into the ion, which is not the reactant for the desired reaction down here. And we can actually see that in our data that the conversion, that when we are dealing with strongly binding sorbents such as primary amines, Ca actually inhibits or has no or even an inhibiting effect on the rate of capture. This is in contrast if we go to the other extreme here and look at weakly binding sorbents like tertiary amines, for instance. In that case, actually the sorbent only carries the proton. The proton comes off here, and that's taken up by the amine, whereas the carbon itself is actually transported as the bicarbonate ion. And hence, in that case, it is a huge advantage to convert to bicarbonate immediately, because that's our final form of the carbonate in the absorption process. And so if we look at this, how does this look in experiments? Here are a couple of early examples of how this was done and how this was illustrated. Perhaps the most simple setup you can imagine is this one. You have a closed container with an amide solution and a headspace, and at time 0, you fill the headspace with pure CO2, and you monitor how the pressure decreases with time. And in this work, they did that for several examples. And for instance, this MEA is a monoethanol amine, which is a popular sorbent. You see that the pressure comes down along this curve here, this grayish curve, and then it becomes flat when the pressure is simply the vapor pressure of the solution. If you add the enzyme here, Ca, you get a slightly better adsorbance. You have a slightly more fast process, which is of course good, but the difference is not big. Now, if you take a weak sorbent, something that binds CO2 very much weaker with a lower enthalpy, then the binding is slower. So the blue curve up here is for MDIA, which is a methyl diethanol amine, and that binds it less efficiently. But when you add the enzyme here in the curve down here, the blue curve, you get the same as if you had the strong sorbent, so that the enzyme sort of removes the difference between the good and the bad sorbent. And that's a big advantage, because the weak sorbent is an advantage when it comes to the desorption, and therefore this is very interesting as a way to compensate the slower adsorption of compounds like MDIA. So more or less the same was seen in another early study here, which is a slightly more technically relevant thing. They used a a spray reactor where the mist of the solvent is lowered through the column while the gas is going the opposite way. And the result here, what you see here is the solvent without and with the enzyme. So the result here again shows that if you have weakly binding solvents such as carbonate ions or MDR that I mentioned before, you get a very distinctive promotion of the rate of absorption when you add the enzyme. Whereas if you have the strongly binding solvents such as MIA, there's no effect of the enzyme. And I think that all has to do with the effect that I was mentioning on the previous slide. Now you can take this one step further and simply add the enzyme to a system for CO2 scrubbing. And this apparently is the original drawing from the patent from 1930 when somebody patented this process, and it's been refined many times since, and it works very well, but it's a pretty primitive process. But it could be interesting to see what happens if you simply add the free enzyme to the solvent that you use in these processes and see if it improves the absorption. And this was done over the last decade or so, and there was particularly a company in Canada, in Quebec, called CO2 Solutions. They developed it quite far, and they built a large demo unit like here. And as you see, they used about 1 molar carbonate as their sorbent solution, and they used enzymes here in quite high concentration. And what they found was that the enzyme promoted the capture even in this pilot scale level. And they developed that further, and not so long ago they were acquired by another company, an Italian engineering company called Saipem, and they have now developed this to the full scale, and they have found a very appealing name for it. They call it the industrial lung, and that's of course because the solvent here is carbonate, and the way it's convert between the different forms is through carbonic anhydrase, and those two are exactly what the processes that goes on in our lungs. So therefore, it's a little bit reminiscent of the process in a lung. I think I don't think there's a full-scale plant built yet by this technology, but I know that Saipem is responding to the tenders that are called, for instance, in Denmark now for CO2 capture for different power plants, etc. Right. So the science of this is to. This is all a good idea. The enzyme seems to help. That's nice. But the science behind this is to develop enzymes that are resistant and sturdy enough to be used under industrial conditions. And that's a real challenge here. And there are two ways to go. And well, there are several ways to go actually. But I'm going to talk about two, and that's enzyme discovery and enzyme engineering. That has been very successful for CA over the last decade. So first, what is that? So enzyme discovery is when you monitor the diversity of enzymes that are naturally occurring. So for instance, here's a phylogenetic tree of one type of enzyme. So the end of all these lines here represents one enzyme, so one sequence of amino acids, and they're all isofunctional. So all of these hundreds and hundreds of enzymes that are defined in this circle have they catalyze the same reaction. They're so-called isoenzymes. They have the same function, but a different sequence. So then the enzyme discovery is to try to look into that sequence space and find an enzyme which is good for your purpose. And that there are many different ways to do that. And actually, machine learning and artificial intelligence has improved this a lot. And so this field is rapidly developing. And usually from a technical point of view, what happens is that researchers find an enzyme that's reasonably good for the desired purpose, and then that's launched as a product, an enzyme product here in the first generation. And now, but those enzymes are usually not good enough for long-term use. They will be competed out by better enzymes over years. So then the next step is to engineer the enzyme, which means to change selected amino acids in different positions so that the enzymes get the properties that you're looking for. This could be higher thermostability or better catalytic efficacy or other parameters. For CA, it's often tolerance to alkaline conditions, which is very important for carbon capture. Right. So these are the two processes, and here are some examples of the first process, enzyme discovery for CA, for carbonic anhydrase. And that's been, like I said, this has been unusually successful. I haven't seen any other area where so many very, very stable enzymes have been discovered in such a short time. And the trick here has been to look at hot places. So what the biotechnologists have done is that they've gone out in nature and found very hot, very thermophile organisms. And the easy thing, or the good thing about CA, is that every organism have them. At least almost every organism have them. So you don't need to worry about whether it's there or not. You just need to worry about finding an organism that tolerates a lot of heat. If you do that, then the CA from that organism will also be very thermostable. And here are some examples from a work some years ago where these bacteria here, they were found in so-called hot, what's it called, white smokers, I think they're called. So thermal vents at the bottom of the ocean, and they are very hot, more than 100 degrees. And in the rim of the vents, there are bacterial communities, and those are examples of that. And indeed, the CA from these organisms were very good and worked very well even at 90 degrees. There's also recently there was a report of archean carbonic anhydrases that were found in volcanic hot springs and on land, and they were also very stable. They could keep half of their activity at 80 degrees for 20, even after 20 degrees at 80 degrees. So again, that's very unusual for an enzyme. And finally, there's an example here of a metagenomic enzyme discovery. And metagenomic means that the researchers go out to a location. In this case, it's a Lokachev field, which is here in the middle of the Atlantic, at the mid-Atlantic ridge. And there's a lot of volcano activity there, so there's a lot of hot places. And the researchers just collect a lot of material there, and then they sequence the genes. They don't grow the organism, they just take the DNA material that's there, and then they sequence it, and they find carbonic anhydrases in that, because they know approximately what the sequence of that enzyme is. And through that, they found here's some that were stable at pH 10 to 105 degrees, 104 degrees, which again is remarkable and a very, very promising candidate for industrial use, because it tolerates both high pH and high temperature. And I think I was going to say something there, but I just jump forward here because I can see my time is flying. So there has also been similar successes in engineering. So the second step of finding better enzymes that I showed in the previous slide. There are many different ways of doing enzyme engineering, and actually all of them have proven quite useful for carbonic anhydrase. There are some people have done so-called de novo design, which is over here. And that means that they have taken a protein that is not an enzyme. It's certainly not a carbonic anhydrase. It has no relationship to the function that we are looking for. But since we know that the carbonic anhydrase involves the coordination of a zinc ion, they put in some histidine residues in this three-helix bundle here, which is very, very thermally stable. And then they could coordinate a zinc ion in here in the middle of the bundle. And that complex was able to hydrate, to catalyze the hydration of carbon dioxide. And the activity of this enzyme was, I think it is 3 orders of magnitude lower than the activity of the natural enzymes. But since it's very stable and small, so it has other advantages. And it could well be that this level of catalysis is good enough to use in actual capture. There's also people doing so-called rational engineering of carbonic anhydrases, and that means that they study very thoroughly the structure and function relationship for the enzyme. And in this particular study, they used computer simulations to find the most flexible regions of the enzyme, and they put in sort of anchors to lodge those regions so that they didn't fluctuate so much when the temperature increased, and that way they could make enzyme. I'm sorry, it's very small, but this curve here is one of their enzyme variants, one of the mutants they have made, and it can tolerate 1 hour at 95 degrees without losing any activity. And then actually at 100 degrees, it loses all activity, but at least it's a very, very stable enzyme. And again, a very promising candidate for industrial applications. Finally, there's a directed evolution. So this is a random approach where you simply you insert mutations randomly in your enzymes, or at least more or less randomly in your enzyme, and then you do that for thousands or hundreds of thousands of different enzymes, and you have a fast screening method to select those that by accident were better, and you do that after one generation, you take out maybe 2 or 5 that are good, and then you repeat the random mutagenesis again, and so in several iterative circles. And that way, you gradually pick better and better enzymes. And again, this was a very successful study that generated enzymes that were very stable and very active even at pH values of 10 or higher. So overall, the discovery and engineering of carbonic anhydrase has been unusually successful when you compare with a similar campaign for other enzymes. Therefore, today we actually have some extremely stable and base-tolerant enzymes. But there are still problems. And I think one of the biggest problem right now is that the enzyme dosage that is needed for effective capture is very high. When you calculate how much enzyme you need to get a good improvement of the capture, the catalytic capacity of that amount of enzymes should exceed what you actually capture by many orders of magnitude. So it's as if there's some problem about how to apply or use the enzyme, and I think that's where the real research in this area is occurring now, is the application of the enzyme. Right. Just to round off, the state of affairs for carbonic anhydrase assisted CO2 capture, as I see it, is that there are very good enzymes around. There has been a lot of success in engineering improved enzymes or discovering improved enzymes, and now we have a very good library of promising candidates. So that's certainly one thing that's in place. There are dozens of reports on how the application of these enzymes accelerates capture from flue gas, and there are also very early reports on how this can be used for direct air capture. Well, some people in Australia showed that if you add this enzyme in little beads that floats on the surface of an algal tank for algal growth, you can actually speed up the growth of the algae, probably because the assimilation of bicarbonate has maybe not rate limiting, but at least it has an influence on the rate. And there are also very early reports on how enzymes can, this is a line here where you see how enzyme-assisted capture can be used simply to drag CO2 out of the air and onto your solvent in indirect air capture. And the main problem right now, as I mentioned, is that the catalytic performance of the enzymes compared to what we know they can do is very poor. So there's something rate limiting in the enzymatic process that we aren't aware of, and I think we need to focus on that to develop the field further. So with that, I'll just thank you for your attention. This is our group here. All this work is done in close collaboration with Novozymes and our sponsors. Thank you.
Thank you very much, Peter, for the nice summary and the talk about the CO2 capture and conversion to carbonic